Good evening. I'd like to just make a few general comments before we begin because we've had a very large number of people register for this webinar, and I'd like to give them all a chance to connect. Just want to say that in perspective, the last 5 years have been especially exciting for me because of the great advances, recent advances in research, especially brain science and in epigenetics and better understandings of methylation that now have enabled us to understand more fully what's going on in the brain and how we can, with natural therapies such as nutrient therapy, to help people that we really were not as able to help in the past. I think I've learned more in the last 5 years than I had in the previous 20. So let's get started. The topics that I'll be focusing on today: nutrient therapy and brain disorders. I want everyone to be apprised of the great power that nutrients can have in helping people with disorders as serious as schizophrenia, bipolar disorder, depression, etc. The next topic, we will be talking a lot about methylation. I've been working with methylation for more than 30 years, and I've now determined and evaluated methylation characteristics of more than 30,000 patients over that time. And it's absolutely a very, very important aspect of clinical work. I mean, one needs to know whether a patient has normal methylation, undermethylation, or in many cases, overmethylation. And then epigenetics, which is a word I imagine most of you are quite familiar with now, but this is something that's come to the forefront in just the last 5 or 10 years. And we've known for quite a while that epigenetics has a lot to do with cancer. And we also know that epigenetics has a lot to do with heart disease. But it's a fairly recent event, more in the last 5 or 6 years, where it's become more and more clear that epigenetics has, in some cases, everything to do with some mental disorders. It now looks like disorders such as schizophrenia, post-traumatic stress, autism may very well be epigenetic in origin. And that means a lot, because in research and in any endeavor, it's hard to fight an enemy or to solve a problem if you don't know who it is. It really helps to know the etiology, the root cause of a condition. And then finally, I'd like to give an example of clinical applications, how we can take this new information on methylation, epigenetics, etc., and then use it to help patients. Our organization is the Walsh Research Institute. We're based in Naperville, Illinois. We are a 501c3 public charity, so we are a nonprofit. Our expertise is limited. We focus on brain disorders, behavior disorders, attention deficit disorder, autism, depression, schizophrenia, bipolar disorder, and Alzheimer's. And really, we don't usually seek patients or help people with other problems. We focus on brain disorders. For the last 11 years, we've been carrying out an international physician training program. We've now trained more than 200 doctors in places like Australia, Norway, Ireland, and more recently the USA. And that's a major part of what our organization does. We also are continually and presently involved in research. Right now we have 4 rather important research projects that are ongoing, 2 of them involving epigenetics. Well, clinical experience, I've been doing this for quite a while. I started out as a you might say, in hard science, and I was working for places like the Institute for Atomic Research and Los Alamos National Laboratory, Argonne National Laboratory, and for many years I really had no research interest or experience in brain biochemistry or in medicine. However, over the last 30 to 35 years, I've now worked directly with 10,000 behavior cases. Most of them people with a problem with control, people, and in many cases of violent children and adults. 5,600 cases of ADHD, 3,500 cases of schizophrenia and bipolar, and that is a lot of cases. I remember many of these patients because their case histories, their medical histories were so extraordinary, and their problems were so great, and their pain was so great. 3,200 cases of depression. I think I have the world's biggest chemistry database for depression, and just about 6 weeks ago, at the annual meeting of the American Psychiatric Association, a colleague of mine, Dr. Robert DeVito, and myself, we presented a paper on depression, trying to make sure that the psychiatric community was aware of the fact that depression is not a single condition, but in fact is really a name given to at least 5 completely different biotypes with completely different neurotransmitter problems and really requiring different treatment approaches. We've also seen 6,500 cases of autism, and up until a couple of years ago, I was pretty sure that myself and my colleagues had seen more autistics than anyone in the world. So from all of this, we've developed a massive chemistry database. I've always been a numbers person. I guess that came from my hard science background, and I've always collected all of the data I could get my hands on. And we've done laboratory testing of 30,000 mental health patients and a lot of controls, because it's important, of course, to know what normal is. I've got more than 3 million chemical test results for blood and urine for patients diagnosed with these disorders. And also really important is it's not just the chemistry, but I learned early on in clinical practice that knowledge of the patient is almost or sometimes equally important as the lab results themselves, because every one of the chemical imbalances we work with has classic symptoms associated with it. So we've also got a couple million medical history factors for these populations. So what have we learned from all this database, from this enormous database of millions of lab results? Well, I guess the bottom line is that there are striking blood and urine chemistry differences between mental illness populations and the rest of society. And believe me, it doesn't take a genius to see it, because these differences are massive. So I wrote a book about a year ago, and I decided to call it Nutrient Power. And the reason I did that is because I think that's one of the barriers to people, whether they're researchers or psychiatrists or other doctors or even the general public. A lot of people just don't believe or don't realize the power that nutrients actually have. Well, it turns out nutrients have a dramatic impact on neurotransmitter synthesis, because many of the key neurotransmitters are created or synthesized all day, every day in the brain. And the raw materials, the ingredients that we need in our brain to make serotonin and dopamine and some of the other neurotransmitters are nutrients, amino acids, vitamins, minerals. So if a person has a genetic or some other imbalance or abnormal levels of one of these key chemicals, one of these key nutrients, you can expect the person is likely to have problems with their mental functioning. We also know, based on recent research, that nutrients have a really powerful effect on gene expression, on the epigenetic regulation of gene expression, and I'm going to get into that quite a bit of detail a little bit later. Another part of this is that we now know that when it comes to depression and anxiety and bipolar and schizophrenia, we know that the key is not quite so much the amount of the neurotransmitter present, but really the more powerful impact is on reuptake processes. That process of reuptake, the process in which a neurotransmitter, once sprayed into a synapse, really how rapidly it returns to that original cell. The pharmaceutical world has learned that, and since the mid-'80s, they realized, for example, with depression, that when they treat depression, they're not trying to increase the amount of serotonin. What they really need to do to make an impact is to affect reuptake. In the case of most depression, they use a reuptake inhibitor. And by the way, that has to do with changing the epigenetics of depression. And then the fourth aspect of nutrients that are extraordinarily extraordinarily important is antioxidant protection. Virtually every condition that I've mentioned so far, every one of these mental disorders and brain problems, virtually all of them are associated with high oxidative stress. So, and it's nutrients that provide antioxidant protection, not just the antioxidants that we might be taking as supplements, but we have natural antioxidants like glutathione and methylation that are really powerful and wonderful in protecting us against oxidative stress. So when you look at all this data, and I've been, of course, staring at it for 30 years, we find that there are some high incidence imbalances in mental disorders. And this goes across the board, really, for all of these different mental disorders. Methylation disorders are really common in mental disorders, and it can be overmethylation or undermethylation. Zinc deficiency is probably the most common, the most frequent imbalance we see in all of these put together. Most of the patients we find who have depression, anxiety, behavior problems, ADHD, etc., are either low normal in zinc or absolutely deficient in zinc. And so many of them improve just by normalizing their zinc. Copper overload is present in a number of conditions. For example, hyperactivity. About 40% of people with ADHD have a copper overload. About more than 90% of autistics have a copper problem, either an overload of copper or of divalent free radical copper. It's really a major problem in postpartum depression. We've done studies and published studies on postpartum depression, and it usually involves extraordinarily high levels of copper. Copper is supposed to be homeostatically regulated in the body. Some people do not have that ability. And in the process of 9 months of gestation of a pregnancy, a woman's copper level more than doubles, and that's what's normal because that's needed for the baby to grow. However, within 24 hours after the pregnancy, after the child's born, the copper level is supposed to go back to normal. But a lot of people don't have that ability. So copper overload can be a cause of striking problems, and I'll get into that a little bit more detail. I'll give you an example. Folate deficiency or folate overload, that turns up over and over and over in a variety of different mental disorders. Pyrrole disorders. Pyrrole disorder is an inborn disorder that has to do with an excessive amount of pyrroles being produced, mostly in your bone marrow and in your spleen. And the net result is that you have a byproduct of important reactions. Are pyrroles, and these pyrroles are just sent out of the body because generally they don't have any real function, nothing good, and they just go out through the bloodstream and out through the urine. Now the problem is that pyrroles have a tremendous strong affinity for zinc and for B6, and they tend to strip zinc and B6 out of the body. So, and then we often find people whose only major problem is a toxic metal overload. It can be mercury, it can be zinc, mercury or lead. There are a number of toxic metals that can cause a lot of problem with brain function. And then fatty acids. There are more than 300 fatty acids in the human body, but really in the brain, at the synapses, at the receptors, there are only 4 of them that are in really high concentration. I think it's between 90 and 95% of all the fats right at where the action is in the brain are in high concentration, and that's EPA and DHA, which are omega-3s, and 2 of the omega-6s, arachidonic acid and DGLA. Now, each of these factors have a really powerful effect on the synthesis of neurotransmitters and a regulation of neurotransmitter activity. These particular imbalances. I was always puzzled early on in clinical work wondering why we kept seeing these very same, just a few of these factors turning up over and over in people with mental problems. And eventually it became clear. See, there are more than 300 important nutrients in the body. And one really fortunate thing, at least when you're working with people with mental problems, is that it's really these 6 factors tend to dominate with respect to imbalances that affect mental functioning. And I kept wondering why such a small number. It's really convenient, by the way, because if we had to do lab testing for 300 or for 100 or more factors, it would be really hard to do. And if we had to have protocols that would normalize each of these, perhaps 100 factors, that would be really difficult. So it's really fortunate that there are only a few that dominate. And after a few years, it dawned on me that the reason that these particular imbalances were the ones that were most important and most critical in helping a person with a mental problem is that these are the ones that have a direct impact on either the synthesis of neurotransmitters or the regulation of neurotransmitter activity. So I'll give you a couple examples. This is the last step in the synthesis of serotonin. And you start with 5-HTP, and then there's an enzyme, a genetically expressed enzyme, and a cofactor of PLP, pyridoxal 5-phosphate, or also known as vitamin B6. It's an active form of vitamin B6. And if a person happens to have significant deficiency of B6, that reaction doesn't go well. And that person is very likely to have less serotonin than required. An example is a Pyrrole disorder. These people are because of this inherited disorder, they are very low in B6. And virtually all of them, at least the ones that I worked with, are low in serotonin level. Another example, this is an example of, shows the synthesis in the brain of norepinephrine. This is the very last step in the synthesis of norepinephrine. And by the way, norepinephrine, serotonin, and dopamine in the brain, all of that is made in the brain. There's a lot of serotonin and dopamine in the rest of the body, but all of these neurotransmitters in your brain are made in your brain exclusively. Well, the last step is all your norepinephrine, which is an important neurotransmitter, comes from dopamine. It happens in little tiny dopamine vesicles in your brain neurons. And the reaction goes because you have a dopamine beta hydroxylase. That's one of the most famous enzymes in biochemistry. And you have vitamin C and oxygen as cofactors, but the main cofactor is divalent copper. And the copper binds to the enzyme, and the enzyme doesn't work well at all if it doesn't have the copper there. And what scientists have learned is that if there are abnormal levels of copper, the difference in dopamine and norepinephrine levels can be massive. There have been two separate studies, animal experiments, where they starved the animals of copper so that they only had 25% of the normal amount of copper in their bloodstream. They then analyzed and determined the amount of norepinephrine and dopamine in their brains. And what they found was by knocking the copper level down to 25% of normal, the ratio of norepinephrine to dopamine could be changed by as much as the factor of 300% or more. And that's a massive change. And dopamine is sort of the feel-good neurotransmitter, whereas norepinephrine is usually considered a excitatory, or if you have too much norepinephrine, you would have a tendency for anxiety, sleep disorder, and in some cases even psychosis. So if a person has really high levels of copper, especially divalent copper, they would tend to be low in dopamine, high in norepinephrine, and we've seen that clinically in thousands of patients. So let's get to the subject of methylation, which is one of our major topics today. First of all, one needs to know that most people have normal methylation. That is a normal amount of available SAMe, a normal capability for methylating other chemicals in the body. 22% are undermethylated, and about 8% are overmethylated. We have seen a lot of the undermethylated and overmethylated patients over the years, thousands of each, really. So, methylation and brain disorders. We've known for a long time that, that this is important, and as I said before, I've personally been involved with a group that has determined methylation status for more than 30,000 patients over 30 years. It was exactly 20 years ago. I went to the American Psychiatric Association and presented a paper informing them and notifying them of the great importance of methylation in mental disorders. Anyway, about a high percent of persons with mental disorders, about 70%, exhibit a serious methylation disorder, far greater than the 22% that are in the general population. For some reason, most people with a methylation disorder don't need treatment and get along quite nicely. But a significant percentage of them run into real trouble. It might be depression or anxiety or even psychosis. And what we've learned is that if we treat the methylation abnormality and normalize methylation, a really high percentage of them improve greatly, and in many cases can throw away their psychiatric meds. Anyway, one thing that we've learned and is absolutely definite is that accurate diagnosis of methylation status is essential for effective treatment. So, methylation disorders, there are two types: undermethylation and there's overmethylation. Now, the incidence of undermethylation can be extremely high in certain disorders. For example, autism spectrum disorders. I had, back in 1999, I had the world's biggest database, chemistry database for autism. And the great Bernie Rimland asked me to, found out about this, and he asked me to present my chemical findings for autism at one of his famous think tanks, where he would have maybe 50 or 60 autism experts from around the world coming together, trying to exchange information and help each other learn how to help these children. Well, what was interesting is I told the group that I found they had a very high level, much higher level of toxics in their system than even their own siblings. Nobody was surprised. They already knew that. I mentioned that many of them were zinc deficient. They all knew that. I mentioned that copper was kind of abnormal for many of them. They knew that. I talked about malabsorption. And then I reported that more than 90%, and I thought maybe more than 95% of autistics were undermethylated. And they all said, what? At that time, methylation and autism had really not been connected. So for a couple of years, I would go to these meetings and we would just continue to discuss methylation. And fortunately, some real methylation experts, especially Jill James and Richard Deff and a few others, got interested in methylation and autism. And now that's a major interest in the diagnosis and treatment of autism, especially treatment. Antisocial personality disorder, 95% of them. I've worked with more than 10,000 behavior disordered people. I used to run a ex-offender program when I was a lot younger. I was a prison volunteer for 18 years when I was quite young. And I got to do a lot of testing of these criminals, and I had a NIH program studying people in prison, and I've tested more than 800 felons. And the antisocial personality disorder, the people who used to be called sociopaths, almost all of them are undermethylated, 95% at least. Schizoaffective disorder, that's a form of schizophrenia. That's a thought disorder in which the primary symptoms are delusions. 90% are undermethylated. So if a patient comes and part of their medical history is that a psychiatrist diagnosed them with schizoaffective disorder, we expect that they're going to flunk our lab test for methylation, and we're going to find that they're undermethylated. And that almost always happens. Oppositional defiant disorder. And we've seen 10,000 cases of behavior disordered children and adults. A lot of them were oppositional defiant young men and women. That is clearly associated with undermethylation. Anorexia, 82%, depression, 38%. Overmethylation. Yes, overmethylation also occurs in a lot of people. It's very high in people who have anxiety or panic attacks. High anxiety is associated with overmethylation. Overmethylation, because of epigenetics, we now understand why overmethylation has this impact. It results in extraordinarily high levels of norepinephrine activity and adrenaline activity, which is why they tend to have sleep problems as well as the anxiety attacks. Paranoid schizophrenia. So this is the highest biotype of schizophrenia. 52% of paranoid schizophrenics are overmethylated. ADHD, 28%. Behavior disorders, 23%. Depression, 18%. And one interesting fact, and one thing we reported at the APA last May, we wanted to make sure that they understood that there were simple blood tests in a medical system where they could identify people who had the overmethylated type of depression and those that were undermethylated. Undermethylated depressants are low in serotonin activity, and they're the ones who tend to do really well on SSRI antidepressants. There are 3 other biotypes of depression that really don't have much to do with serotonin at all. And then there's this one, overmethylated depression, 18%. These are the ones who get worse if they get an antidepressant. And the reason is they already have too much serotonin activity. And these are the people who might develop homicidal or suicidal ideation after taking an antidepressant. And there have been studies recently that show that a very high percentage of school shootings are related to young men or women who were okay, actually, did not really have a serious behavior problem until they were maybe 14, 15, or 16 years old, developed anxiety and depression, and were given an antidepressant, and then got dramatically worse, and then disaster happened. And there's just case after case of that. In fact, we've looked at the last 50 major cases of school shootings since year 1990, and more than 40 of them involved antidepressants and dramatic adverse reactions. Okay, so let's look at undermethylation now. What's the cause of it? Why are people undermethylated? So many of them. Well, the answer is most of them get this genetically, and it's inherited, and this involves enzyme mutations that have built up over thousands of years. Enzyme mutations called SNPs, single nucleotide polymorphisms. And there are several enzymes in the methylation cycle, and these SNPs result in some level of weakening of the enzymes. And most of you have heard of MTHFR, and there's MS, and there's about 8 primary enzymes in the methylation cycle. There are some lesser common causes of undermethylation or factors that tend to make it even worse. That is, histamine overload can do that. And if a person has protein deficiency or malabsorption, then the amount of methionine that makes it into your body can be too low, and that itself can cause some level of undermethylation. But primarily, it's the enzyme mutation, the SNPs in the cycle. Well, this is a picture of the sort of a summary of the methylation cycle. I didn't put everything in. I didn't want to clutter up the diagram, so there are a few things that are missing. The green ovals are some of the enzymes, most of the enzymes. They're not all in here. Again, I didn't want to clutter up the diagram totally. But basically, this is the methylation cycle, and this is where our methyl comes from. Methyl comes originally from the diet, protein containing methionine, sort of the beginning, and that's where the methyl group first comes. And this entire cycle is, the very first part of the cycle involves methionine creating a relatively unstable molecule called SAMe that delivers methyl. But this complex cycle has the objective of keeping methylation levels normalized, and these cycles are constantly spinning. These reactions that are shown here in these circles are just constantly spinning and spinning. There's more than 1 million methylation reactions per second in a typical person. So it's sort of like the Indianapolis Speedway, where you've got these cars constantly circling around and circling around. This goes on constantly. One interesting fact is that ATP, adenosine triphosphate, which is up in the upper right corner there, that is a chemical that provides energy for the initial part of this reaction. All of the ATP cycles in the body are so great that this relatively tiny molecule, every day on the average, we create more ATP than our weight in pounds. Kind of amazing. So let's just go through this really quickly, and I know many of you know this completely, and maybe a few of you are not quite so familiar, so I'll go through this rather quickly. The first part of this is your methionine from your diet, one of the proteins in your diet, reacts to form SAMe, and the enzyme there is shown, MAT enzyme, and magnesium and ATP are needed for that to go. And this is how you create the fairly unstable molecule called SAMe, and that's the primary donor, methyl donor, throughout the body for methionine, for methyl groups. So then you have the methyl donation. The SAMe finds some other biochemical that it donates a methyl group to, and then the SAMe minus the CH3, which is a methyl group, leaves you with acetinosyl homocysteine. A very interesting chemical, by the way. Acetinosyl homocysteine itself is known to have epigenetic characteristics that were not quite well known until a couple of years ago. So then the next step along the cycle, is your acetinosyl homocysteine then is converted to homocysteine. In this case, you need the assistance of 3 enzymes. The reason you need the 2 at the lower right is that if you just had SAH and homocysteine together, thermodynamically that goes back to SAH and not homocysteine. So the only way this cycle can go is to take away the adenosine, to have it disappear or to be metabolized, and the enzymes that cause these reactions to remove the adenosine are shown. Then you have the glutathione pathway. Part of the homocysteine goes through a pathway that sort of leaves the methylation cycle and does a really important thing, namely it creates some of these really important sulfur-bearing antioxidants. Starts with cystathione. It's called the cystathione pathway, cysteine, and then glutathione. And again, there is an enzyme called CDS, and the homocysteine reacts directly with serine. That's another amino acid. And B6, again, is a cofactor. Well, there's 2 ways that homocysteine can make it back to methionine, because the cycle has to go back to where it started. And so what you have to do is put a methyl group back on. So there are 2 ways in which a methyl group can be put on the homocysteine, which then becomes methionine. One of these is this pathway that some people call the TMG remethylation. TMG is trimethylglycine. And trimethylglycine can react directly with homocysteine with the help of the BHMT enzyme, and it forms methionine. And DMG. DMG is dimethylglycine, so it's trimethylglycine that's lost the methyl. Now, for many years, this was considered to be of relatively minor importance compared to the folate remethylation cycle. But in the last 2 years, we found out something rather surprising. Recent research shows that the amount of TMG in the liver is much higher than previously realized. And so people are now beginning to think that this pathway is a lot more important than previously acknowledged. And then we have this part of the cycle called folate cycle remethylation. This is the second way in which homocysteine can make it back to methionine. And there are actually a couple of enzymes at that B12 step. Really, the way it happens is that methyl has to be put on B12 to form methyl B12. The methyl B12 then, with the aid of the MS enzyme, can convert the homocysteine back to methionine. Should be ready to go around the cycle again. But the important feature of this cycle is the part involving MTHFR. And the reason is that the rate-limiting step in this entire cycle is the MTHFR reaction. MTHFR is an enzyme, a genetically expressed enzyme, and this reaction doesn't go without that enzyme. Now, there's been a lot of conversation and a lot of work lately focusing on MTHFR because it is the rate-limiting step. It is the most important of the enzymes in this entire methylation cycle. Well, MTHFR is a very large enzyme. It has more than 500 amino acids. Its molecular weight is something like 77,000 or 77 Daltons for those of you who are physicists. So what does it mean if you've got a SNP or a mutation in MTHFR? A SNP means that out of those 500 or so amino acids that make up this complex enzyme, means that one of those amino acids is in the wrong place, or is the wrong amino. In other words, out of this complex array of 500 amino acids that make up MTHFR, 499 of them are exactly what they're supposed to be, but one of them is something different. You might have an arginine where methionine is supposed to be, for example, or glycine. Okay, so again, this is the total methylation cycle, and it can be thought of sort of like a biochemical engine for making SAMe, which can then donate the CH3. So that explains. Now, if you have a SNP in any of these enzymes, there's a possibility that the enzyme will be weakened. Now, most SNPs do not have any significant effect on the function of an enzyme, partly because the enzymes are so large, and you're only changing just a tiny part, only a single amino acid in that really large enzyme. But there are exceptions. For example, the 677T location along that MTHFR enzyme is one that is in a critical location. So if you happen to have MTHFR SNP, especially if both strands of your DNA, especially if it's what's known as homozygous, where you have two of these SNPs at the same place, at the 677T, then you can have a significant weakening in that enzyme, and the whole cycle can sort of shut down. So let's move on. Well, that explains methylation and why people can be undermethylated. It's almost always due to SNPs or weakening in these enzymes that enable that methylation cycle to function. Well, how about overmethylation? Well, that happens mainly in the pathways of utilization of SAMe. And what happens to the SAMe once this methylation cycle is putting out all this SAMe available for reactions? Well, an interesting thing is that there are more than 80 really important methylation reactions in the body. And some of them are just absolutely essential to life. But more than half of all that SAMe goes to a single reaction, and that is the synthesis of creatine. Most people think that it's about 70% of all the SAMe in typical people goes to creatine. It varies somewhat from person to person. It can be as low as 40%, as high as 80%. But the lion's share of your SAMe goes to one reaction, and that is the synthesis of creatine. The other 30% goes to other reactions, some of which are extraordinarily important. So how does overmethylation occur? Well, that happens if you have SNPs in the pathway that creates creatine. The reaction is shown in the slide, and you start with arginine and glycine, which are two of the amino acids. And then with a very important enzyme called AGAT, it gets converted to a chemical called guanidinoacetate. Well, guanidinoacetate is the chemical that has the lion's share of the SAMe reacted. And so SAMe plus the guanidinoacetate again, with the aid of a different enzyme, creates SAH. The SAMe gets converted to SAH. The methyl group has been delivered to the guanidinoyl acetate, and you wind up with creatine. So if you have a weakness, if you have a SNP in one of these two, then that can cause overmethylation. You can have a glut of SAMe that's sort of floating around and looking for something to methylate. So to summarize this, the primary causes of overmethylation, number one is impaired creatine synthesis in one of these enzymes, or sometimes arginine or glycine deficiency. Arginine deficiency is fairly common. I did a study of a lot of professional athletes and Olympic people, and I found that arginine deficiency was common in really tall people. It has a lot to do with growth, and working with a number of 7-foot-plus basketball players, I found that a lot of times they had arginine deficiency. They also tended to have not much muscle development, and so you had these really tall people that look like sticks. I call it the minute bull syndrome. And anyway, they had impaired creatine synthesis, inability to develop good muscles. And then impaired cystathione synthesis can also tend to give more methylation than you would like, or normally would have. And then some of these methyltransferases have SNPs. That is the other 80 reactions. A lot of these methyltransferases are the same methyltransferase, or very allied, and so they also can have SNPs. So you can have a lot of these reactions don't go well, and that can cause overmethylation. So what does that mean? Well, it's really important for a clinical patient to know what their methylation status is. You need to know, are they overmethylated? Are they undermethylated? There's been a lot of attempts lately to use genetic testing to try to diagnose methylation. Most of it is focusing on MTHFR and SNPs in the methylation cycle that can cause undermethylation. But the reality is, there's a methylation tug of war. And you have enzymes such as MTHFR, SAHH, and several others, more than the 5 shown, tend toward undermethylation. And then you have other SNPs in enzymes that are tending toward overmethylation, and it's a tug of war. And so that really complicates matters. So how do you do a lab test? We really need to know whether a patient is overmethylated or undermethylated, because that has a lot to do with the direction our treatment's going to go. And it has a lot to do with trying to evaluate which neurotransmitters have low activity or excessive activity or normal activity. Well, the two methods that there are, fortunately, a couple of lab tests that do this overall evaluation that can directly look for the net result of that tug of war of the SNPs. And SAMe to SAH ratio, that is now a lab test that's now routine. It was originated by Doctors Data with the help of the assistance of Dr. Jill James. And then for many years we've used whole blood histamine. And that's a very effective methylation marker. And these sort of take the net effect of all the SNPs that can enhance or can depress methylation. Present genetic tests for MTHFR, etc. I think they're very interesting. They're relatively inexpensive. I believe they're highly accurate, but I think they're not totally reliable in determining whether a person is overmethylated or undermethylated. And we know there are people who have the MTHFR 677T and the 1298, even homozygous MTHFR, who are normal in methylation, and in a few cases overmethylated, which might surprise some people. But it really has to do with which SNPs are winning that tug of war. So fortunately, we do have a couple of blood tests that can determine overall net methylation status and then guide us toward treatment of the patient. And both of these methods, number 1 and 2 both, have some, they're not perfect analyses. And I look forward to a time when we can perhaps even get a better way of doing this. Eventually, genetic testing might provide this. The problem with genetic testing, one problem, is that these SNPs are more qualitative than quantitative. And so even though we know that we might have a powerful tendency, it's hard to put a percent on that because a given SNP will have a different impact on one person compared to another. Okay, so one thing that really helps in diagnosis of methylation status is that undermethylation has a really clear set of symptoms and traits that go with undermethylation. So in diagnosing methylation, we need not just the lab results, which are really helpful, but it really helps if you have it coinciding with these key symptoms and traits. For example, undermethylated people, very strong-willed, usually from birth. Their mothers will say that they saw this by the time the child was 3 or 4 years old. If they get into sports, they tend to be very competitive. They want to win. They do their best. I've had a number of long-distance runners come to our clinic for depression or other problems, and if they were, I always want to know, were they competitive? Did they practice? Were they constantly trying to improve? If the answer is yes, you could be about 90% sure that they're undermethylated. Obsessive-compulsive tendencies are part and parcel of undermethylation. And seasonal allergies, 75% of undermethylated persons have seasonal allergies, problems with ragweed, grasses, things like that. Addictive tendencies. Undermethylated people need to be especially careful about alcohol and illegal drugs because they have a tendency to get hooked on things far more rapidly than others. For example, the average person who abuses cocaine, let's say they snort cocaine recreationally, which of course is a terrible idea, but the average person, typical person, it takes roughly, on the average, they believe it takes about 2 years before they get really addicted, totally addicted to cocaine. But if a person is severely undermethylated, they can get addicted the very first time. And I've met people who were very undermethylated who told me that they got completely hooked on cocaine with the first time they snorted it. Another interesting aspect is high libido. People with undermethylation, whether they're male or female, have a high sex drive. It's just part and parcel of it. They have a family history of high accomplishment. I think the strong will, the tendency to be perfectionist and to be good at things they care about and to win tends to be, they tend to have high accomplishment and at least a family history. And if I took if you took 100 psychiatrists or 100 CEOs or 100 great athletes, even 100 doctors, you would probably find that 80 or 90% of them were undermethylated. Calm demeanor, but high inner tension. These are people who might have high anxiety and it just doesn't show. But a lot of people experience that, and that's one of the clear symptoms and traits of undermethylation. High fluidity is another example. People who are undermethylated have much more tears and saliva than average. If you're walking down the street and you see somebody spitting on the sidewalk, it might be disgusting, but he also might have an undermethylation condition and just an excessive amount of it. Non-compliance with therapies. People who are undermethylated cause all kinds of problems with medical practitioners because they tend to be non-compliant. They just don't like to comply with any therapies. I've met a number of undermethylated people who told me that they had severe headaches. They knew that aspirin would help them, but they wouldn't take aspirin for the headaches. It's just part of being undermethylated. Now, this is a list of 10 symptoms. In my book, Nutrient Power, I think we have a list of at least 24 symptoms, so this is just a partial list. But after meeting a patient and doing a careful medical history, we are usually very accurate in identifying whether a person is undermethylated, and we can predict with a lot of accuracy the lab results. So similarly, overmethylation has classic distinctive symptoms and traits. Now there's exceptions. These are generalities with many exceptions, but if a person has, say, 30 or 40 or 50% of these characteristics, you can be pretty sure they have that methylation disorder. Overmethylation is very different. These people generally, almost all of them, have high anxiety, very high incidence of sleep disorder. They're high energy, their bodies move a lot. Verbosity, they tend to talk a lot, and they have, if a surprising number have high artistic and musical ability. I've worked with hundreds of people who were professional artists or musicians, and so many of them were overmethylated. Overmethylation, by the way, has a lot to do with excessive activity or unusually high activity of neurotransmitters like dopamine and norepinephrine and serotonin, and we often wondered if maybe it was this additional neurotransmitter activity that had something to do with their musical and artistic abilities. Most people who are overmethylated are intolerant to antihistamines. They just feel anxious or nervous or just unwell if they take an antihistamine, as opposed to the undermethylated people who usually thrive on antihistamines. Overmethylated people have high empathy for other people. They make wonderful neighbors. They often care about other people more than themselves. Many of them volunteer to charities and give of themselves. They're really quite inspirational people. However, the undermethylation may cause high anxiety and in some cases a very nasty form of depression or other mental illness. I think about 15% of overmethylated people get into trouble. The other 85% for some reason sail through life and don't need treatment. But if they are having trouble with anxiety or depression or even schizophrenia or bipolar, and if we give them a treatment to normalize their methylation, most of them get a lot better. So, low libido, and these people tend to have dry eyes and dry mouths. It's not a good thing if a overmethylated and undermethylated person marries each other, because if they have the difference in libido, it could cause obvious problems. People with overmethylation often are not able to wear contact lenses because their eyes are just too dry to support that. If they take an SSRI or SAMI or methionine, they usually get dramatically worse, especially if they have anxiety or depression. In this case, the SSRIs or even SAMI usually make their depression and anxiety much worse. And they tend to have low blood histamine and elevated SAMI to SAH ratio, which is the blood part of it. Now, when I was looking at this and preparing this slide, actually yesterday, I heard about Robin Williams, who had just killed himself. And if you look at Robin Williams and if you look at these characteristics, the high energy, the verbosity, the artistic ability to be an actor, etc., high empathy for others, he certainly had that. He was not a competitive person. I sort of wonder whether or not he was unfortunately recently given a new antidepressant, an SSRI, and had this adverse reaction, because we know that SSRIs for a small number of people can cause suicidal ideation. I'm hoping to find out sometime if they'll release what medication he's on and if it is a new one, because that might have been why he killed himself. It's very possible. Impact of methylation disorders. We'll say a person has overmethylation or undermethylation. And again, that's generally inherited, runs in families. The greatest harm actually happens before you're born. The greatest impact of methylation is altered DNA methylation. That's a primary aspect of epigenetics. And for those for whom that is new, I'll give you a summary of that in the next 5 minutes. So during early fetal development, your DNA is regulated by methylation, which by SAMI, that's the donor, and you need to have the methyl on the right parts of the DNA because that helps regulate DNA and genetic expression. And so if you've got a abnormality, abnormal methylation, it could give you a predisposition for cancer, a predisposition for depression, mental illness, definitely autism. Also, gene regulation problems are associated with altered methylation of protein aggregates called histones, and I'll describe those, and I'll even show you a picture of that in a couple of minutes. And then you also have altered kinetics of more than 20 essential methylation reactions. In some cases, too little, other cases too much, depending with whether you're over or under methylated. So I want to say a little bit about single nucleotide polymorphisms. We share 99.9% of our DNA with everybody of the same gender, and it's that 0.1% that really makes us very different. And this has developed over the years. I think humans were much more similar, if you believe that they existed hundreds of thousands of years ago, and as these mutations develop over time, you get these differences. So what are SNPs? SNPs are gene mutations that developed over thousands of years. If you do a genetic test and you find out you have a lot of SNPs, don't be surprised. They've recently counted up all the SNPs that have been identified in the human genome, and the last time I looked, the number was at 10 million. There are more than 10 million SNPs, or mutations, that have already been identified in the human genome. Most humans have more than 1,000 SNPs, and a lot of people have 10,000. So having SNPs is not necessarily an unusual thing at all. I really doubt if there's any human beings alive that don't have SNPs. So enzyme SNPs, these are the ones that can be most important. They're more likely in very large enzymes, and that's just based on statistics alone, I think, a lot of it. And it turns out that most SNPs have little or no effect on enzyme functions, but some strategically placed SNPs can significantly weaken, not destroy, but weaken enzyme function. MTHFR 677T is a good example. 1298 can, to a lesser degree, also do the same thing. But the impact of the SNP varies from person to person. Okay, so methylation and mental health. Now we need to get into the subject of epigenetics, and really, we really should not be considering methylation in isolation and epigenetics in isolation. The two of them are just intertwined. They're just intrinsically intertwined. Methylation is a dominant factor in epigenetic processes. SAMe, folates, niacin, other nutrients have a powerful epigenetic impact on neurotransmitter activity of synapses. And I'll explain the mechanism in about 2 minutes. So that's extraordinarily important. And more than 60% of ADHD, anxiety, depression, and even psychosis patients exhibit a serious methylation disorder. That is a lot of people and a lot of human misery. And it looks like methylation imbalances are, in some cases, the dominant factor in those disorders. So, epigenetics. What is epigenetics? We have more than 20,000 genes in every cell in our body. 20,000 genes in our DNA. A typical person has about 1 trillion cells. Every cell contains a small element that is a nucleus. And inside the nucleus is your DNA. And some of your DNA is with you from birth, and you have the same identical DNA molecule, especially in your brain, the same DNA that you were born with. And interestingly, the DNA actually moves. It sort of breathes back and forth in this tiny little fragile molecule. It's amazing that it doesn't tend to break apart and stop functioning. Well, the problem is we have the same DNA in every part of your body. However, different parts of your body, like your liver, your skin, your brain, other tissues, you require a unique combination of proteins. Well, what do genes do? What is a gene, and what does it do? Well, a gene has one job, and only one job, and that's to make one specific protein. So you've got the possibility of thousands, a lot more than 10,000 genes or 10,000 proteins that might actually be expressed or appear in different parts of your body. But you need to have just the right chemicals in every part of your body. You need to shut off a lot of these protein producers tissue by tissue, and even in the brain location by brain location. So how's this done? It's done by epigenetics. Epigenetics is how these genes are regulated, how some of the unwanted genes, say in your liver, are prevented from forming these unwanted proteins. And the way it happens is by methylation primarily. during pregnancy, chemical bookmarks, methyl marks, attached to DNA, and in general, if it doesn't attach to the DNA, it tends to enhance gene expression. If you do have methylation, in most cases, in general, it tends to inhibit or shut down gene expression, or say it a different way, shut down the production of that particular protein. Now, it turns out that environmental insults at any age, just took a drink of water, environmental insults at any age can alter gene bookmarks. It can happen in utero and can cause all kinds of problems, including birth defects. It can also produce mental disorders or other disease conditions. So, what are the characteristics of an epigenetic disorder? It now looks like there are a number of major disorders like post-traumatic stress, schizophrenia, autism. There's more and more evidence that, in fact, they are epigenetic in nature, that it's a major part of their etiology. So what are the characteristics? What are the clues that something might be epigenetic in nature? Cases of sudden onset after normalcy. For example, post-traumatic stress. Another example is perhaps a schizophrenia mental breakdown. Another example might be regressive autism. So that's number one. And then persistence of the condition after onset. It doesn't go away once it comes. It comes suddenly and then be with you. It can be a problem in many cases the rest of your life. A multitude of characteristic symptoms, especially symptoms that suddenly appear. For example, in regressive autism, you might have a child that loses speech, might develop odd characteristic movements. They might lose eye contact. They might have divergent gaze. They might have immune problems that weren't there before. They may have food sensitivities that weren't there before. And the reason that you would expect that is that epigenetic disorders are caused by environmental insults. And if you've got a sufficient insult to change one gene, you're probably going to change a dozen genes or maybe 100 genes. So a characteristic of an epigenetic disorder is that characteristic symptoms, numerous ones, are usually present. Another thing is that epigenetic disorders are often heritable, meaning they run in families, but they violate laws of genetics, the classic Mendelian laws of genetics. Another thing is that you often see abnormal methylation. And another factor that is not mentioned too often, except in people who are doing cancer research, is almost always you have severe oxidative overload. And it now looks from the research that's been going on recently that the most common cause of an epigenetic disorder, which really is altering one of those methyl marks, and usually a methyl appearing where it's not supposed to appear on the DNA, that shuts off a protein. It usually has a lot to do with overwhelming oxidative stress. So I've sort of looked through all the disorders and looked at these criteria, and it looks quite clear to me that schizophrenia is likely to be a gene regulation disorder, to be epigenetic in nature. A couple of years ago, I published a theory of schizophrenia, and I'm right now organizing a study to try to prove it, to demonstrate that it is. And I'm connecting with a very capable genetic group, research group in Australia, and we hope that we are about to begin a classic experiment looking at these methylation abnormalities, methyls in the wrong location along your DNA strand that occur for schizophrenia. And we're doing it for the different types of schizophrenia, because there are 3 different kinds of schizophrenia. Bipolar disorder also very often involves a breakdown. I've worked with more than 1,600 cases of bipolar, and I think 1,200 of these cases at least talked about how the bipolar came on rather suddenly, or at least there was a dramatic worsening, or what you might call a breakdown, that happened very often between the ages of maybe 16 and 26. Post-traumatic stress disorder fits all of the characteristics of an epigenetic disorder. people who were okay before an event, which can be either emotional or it can be physical. It makes a change that, it starts a disorder that doesn't go away. Wilson's disease, autism. About 3 years ago, I gave a model for autism that I think fits what autism really is better than any other theory I've ever seen, and that basically involves an epigenetic environmental insult that causes epigenetic changes, methylation of either the DNA itself or the proteins near it, and that is what I think what causes autism. And there's been recent evidence, recent research, published research, that is making it look more and more likely all the time. Obsessive compulsive disorder. And the one thing we're really certain of is that cancer researchers now are convinced that most cases of cancer are epigenetic in origin. And I think 70 or 80% of all cancer research now is aimed at epigenetic therapies, trying to find ways to either cope with the gene regulation that's gone wrong, or finding ways to normalize those bookmarks, those methylation marks that are now deviant. In cancer, they've also learned that what nearly always happens is that it has to do with methylation occurring where it's not supposed to occur. And what that does is it tends to shut down a protein. And in the case of cancer, shut down cancer prevention proteins. And they've even identified the genes that are awry and the proteins that are absent that are needed to cope with cancer condition. Heart disease is also pretty well recognized. Many forms of heart disease appear to be epigenetic. That is, that they caused by either cumulative or a sudden environmental insult. Well, there are two epigenetic processes that are completely different. Number one is the one I mentioned earlier, the DNA methylation that's established in the womb, and this has to go right so you get the right chemical mix in every part of your body. And then there's also histone modification. Histones are proteins that the DNA wraps around in the nucleus. The DNA isn't just all jammed together. It wraps around these little balls of histone proteins. So let's look at the first process of epigenetics. That's DNA methylation. It's established in the womb. And what happens, cytosine is one of the 4 nucleotides. And methylation that affects gene expression usually occurs in places called CpG island clusters. And most of that is in either promoter or intron regions of your DNA. And the CpG island is a cytosine connected to guanine. That's another one of the nucleotides with a phosphorus connecting them. And most CpG in your DNA, about 60 or 70 or 80% of it is already methylated, and it's supposed to be. However, there are clusters called CpG islands that may or may not be methylated, and that has a lot to do whether a protein will be produced or not from the corresponding gene. And these early methyl bookmarks usually remain in place throughout a lifetime. It wasn't too long ago, I think 15 years ago, it was believed that these methyl marks were in there like concrete and could never change. But now recently we know there are methylases and demethylases in particular that can actually, the methyl bookmarks can be removed from your DNA, and that's what a lot of cancer researchers are focusing on now, trying to remove methyl marks that aren't supposed to be in a particular place. Well, in utero environmental insults during pregnancy, there are a number of insults that can produce deviant bookmarks and produce an epigenetic disorder and can produce a serious condition. It can be physical or mental, or birth defects. One example we all know there are medications that can produce birth defects, and pregnant women need to avoid them. Anyway, the one unfortunate aspect of this is that a lot of these bookmarks, a lot of these methyl bookmarks, are formed in the first month of pregnancy. And I know a lot of women who, as soon as they found out they were pregnant, they started doing very healthy things. They had a better diet. Maybe they stopped smoking or drinking alcohol and had a healthier diet. Unfortunately, a lot of these problems, a lot of these epigenetic things, it may be too late. A lot of them important gene expression regulations already established before the woman even knows she's pregnant. So really, if they want to be absolutely do this right, they needed to get very healthy and do all those good things before they get pregnant. Anyway, after a person is born, you can still have a severe environmental insult may alter one or more of these gene regulation marks and produce an epigenetic disorder such as cancer or a mental illness. And the insults can be they can be emotional or they can be physical. And this is just a picture of DNA methylation. In this case, it shows the bright lights are methyls that happen to attach to this tiny piece of DNA strand. There are billions of these of each of these cytosine molecules that make up your DNA. And your DNA, by the way, if you were to stretch it out, it would be about 6 feet long. Well, histones. This has to do with the second way in which epigenetics can affect you and the way we can. And this is really important for treatment because histones can be modified. What are histones? They're support structures for the fragile DNA. Your DNA wraps around histones. You've got millions and millions and millions of histones in every cell in the nucleus in your body. And all of your DNA wraps around these proteins. A histone is composed of 8 linear proteins, twisted together like a ball of yarn. Originally, they were believed to serve only as structural support for DNA packaging. Later, it's found that you could have inhibition or promotion of gene expression depending on chemical reactions at histone tails, the parts of these linear proteins that stick, that say sort of got out of or extended from the sort of the ball of yarn kind of approach. And we now know that nutrient therapies and also drug therapies can modify the histones that control reuptake of serotonin, dopamine, and other neurotransmitters. So this is the approach that all present therapies are aiming at in terms of trying to adjust epigenetics and adjust, in our case, neurotransmitter function. So here's a picture of a histone with the DNA wrapped around it. I think it goes around 1.4, 1.6 times in every one of these, and this is what's known as chromatin. Chromatin is basically the DNA and these proteins. So there are 2 main parts of the epigenetic code. One is the DNA methylation that happens before birth, where these bookmarks, these methylation, are attached to parts of your double helix, and can either turn on or turn off gene expression at those locations. And then you have these histone tails, and you can see the let me go back one. You can see these histone tails that stick out of the histone, and reaction in certain locations there has a lot to do with whether a gene will express or not express. So this shows the histones spread apart, but sometimes they can be all jammed together. So what causes a gene to express or not to express, or to have a particular rate of expression or rate of that protein's production? Very often it's a competition between methyl and acetyl groups attaching to the histones, to these histone tails. Competition between acetyl and methyl groups often determines if a gene is expressed or silenced. turns out that acetyl marks on the on these histone tails tends to promote gene expression. Methyl bookmarks tend to suppress expression. Nutrient therapy can change methyl-acetyl ratios and adjust production of proteins and enzymes, and including those that control serotonin and dopamine neurotransmission rates and reuptake. Well, how does this work? For a gene to express, the gene has to uncoil from the DNA. It has to be exposed. The reason is that gene expression requires a direct interaction of RNA polymerase, which is a fairly large molecule, and transcription factors. These are other large molecules, and all of these are genetically expressed, but these are large molecules that have to directly interact with DNA. These large molecules cannot gain access to DNA histone regions if they're densely compacted. They have to get at the DNA itself. You can think of your RNA polymerase as a molecule that is swimming around in the cytosol inside your nucleus of a cell, looking for a gene to express, and it's looking for a gene that has been uncoiled and is available. Now, how does this work? Well, the gentle attachment of DNA to histones involves electrostatic attraction. DNA is a weak acid, ribonucleic acid, etc., and histones are strong bases. The pH is about 7. So that's how they attach. Well, it turns out that if a histone tail is acetylated, if you get an acetyl group, that decreases the histone pH, so you don't have as much attraction, and it causes uncoiling of the DNA. Methylation increases the histone pH, makes it more basic, and therefore causes this whole DNA histone complex to compact. So here's a figure that sort of shows that. So this is a case where methyl has won the war, and acetyl has won the war in this case. You have low methylation or undermethylation, and acetyl has won the war at the chromosome level and reacting with the histone tails. And so what's happened is you have open chromatin, and the genes that are exposed in those areas are available for genetic expression. And this is the opposite. This is when methyl wins the war. It all jams together. It condenses, is a term they use, and so the genes that are within there are not exposed. These large molecules, RNA polymerase, and the transcription factors can't do their job. They can't get there, so nothing happens. So those particular genes are silenced. So that's really how this all works. Now, in mental health, when you have a problem with your neurotransmitters, for a while back in the 60s and in the 70s, most researchers were focusing on how much serotonin is present, or how much are you synthesizing, and how much, and they were looking at the enzymes and the amount of the neurotransmitter. Well, in the mid 80s, brain researchers found out that the real dominant effect has to do with reuptake, the rate at which neurotransmitters straight into a synapse return to the original cell, and that has a lot to do with the population of what are known as transport proteins, or reuptake transport proteins. These are proteins that are in the presynaptic cell. That is the cell that from whence the serotonin or the dopamine originally came from. And these are transmembrane proteins that snake in and out of the protein, and by a couple of mechanisms they can cause the neurotransmitter, which might be serotonin, to very quickly go back where it came from. And that's the dominant factor in neurotransmission of these key neurotransmitters. So these transmembrane proteins, they can remove neurotransmitters from the synapse. This process is called reuptake, and it's like a vacuum cleaner inhaling dust particles. And it can happen really quickly. There are brain cells that can fire several times a second. So you can imagine how quickly the neurotransmitter is ejected in the synapse and then very quickly comes back. Anyway, these proteins are formed by gene expression. The amount of these reuptake proteins, whether it's serotonin or dopamine or norepinephrine, it depends on the methyl-acetyl competition at specific DNA regions. And that means that with nutrient therapy now we have a handle on, and we can adjust neurotransmitter activity in a very powerful way with nutrients. Well, methyl and acetyl are the two most important chemical groups that are related to this histone modification. Well, where does the acetyl come from? Well, the donor for acetyl groups is acetyl coenzyme A. That's a chemical that's everywhere in the body in high concentration throughout every cell in your body. And in fact, any form of food you eat will eventually convert into some acetyl coenzyme A. Whether it's protein can convert into it, fats can convert into it, and so can carbohydrates. Sort of all roads lead to Rome, or all lead to at least some acetyl coenzyme A. And SAMe, we know where that comes from. That comes from the methylation cycle. Well, these are the donors. These are the donors onto those histones. But it turns out that their concentrations in brain cells are relatively unimportant. If you really want to get a handle on say, serotonin activity, you might be able to do some good, perhaps by taking tryptophan or some of the precursors of the neurotransmitters, or try to juggle with your methylation or other biochemical cycles to try to increase the amount of the neurotransmitter if you've got low activity. But that's relatively unimportant and relatively ineffective. What really affects it are enzymes. Four families of enzymes: acetylases, deacetylases, which are probably the most important of these four, methylases, and demethylases. These dominate the attachment or removal of acetyl or methyl groups. So epigenetic nutrient therapy for adjustment of serotonin or dopamine activity has to concentrate on the enzymes. Let's give an example, which some of you I think will find kind of interesting. Niacin, or B3, it inactivates a major deacetylase inhibitor called sirtuin, S-I-R-T-U-I-N. And what this does is it increases expression of these transport proteins, and therefore reduces neurotransmission, because you've got the neurotransmitters are leaving too quickly. Now, in roughly around 1950, the great Abram Hoffer was the first to discover that nutrients could have a massive effect on mental health. And he found that he could very often make great improvements in schizophrenics by giving them niacin. Well, he had a theory that he called the adenochrome theory of schizophrenia, which is a very logical and was the best theory, probably the very best theory that was possible at that time. But now we know what really caused it. Niacin is a major deacetylase inhibitor, and what that means is it can knock dopamine activity down. Schizophrenia is for many years, for about 30 years, has been considered a high dopamine condition, high dopamine activity. Well, niacin knocks it down by this epigenetic mechanism. It's really too bad that Abram died before this was discovered. He would have found this really interesting, and it would have validated what he found, namely that niacin can really help certain schizophrenics. So what are some of the insights? This is a very complex issue. The whole field of epigenetics is still in its infancy, and the amount of knowledge coming out even month by month in the literature is quite astonishing. And it's got a long way to go. We've only scratched the surface. But we already know some things that can already be used in helping our patients. For example, niacin and niacinamide act as dopamine reuptake promoters, which is terrific if you have excessive dopamine activity, which can happen in anxiety. It can happen in some forms of depression. It can also, and of course, also in bipolar and in schizophrenia. We now know that methionine and especially SAMe are natural serotonin reuptake inhibitors. Well, that's what antidepressants are. That's what Prozac and Paxil and Cerazone and all those medications are. They are serotonin reuptake inhibitors. The medications, the drugs, the antidepressant drugs, act very rapidly. They get into your body rather rapidly, get into your brain rather rapidly, and what they do is they impede the connection of the neurotransmitter with these transport proteins. They don't quite disable the transport proteins, but they prevent them from getting causing them to go back into the original cell. They inhibit reuptake. Well, methionine and SAMe, over-the-counter nutrients supplements that can do the very same thing. The difference is that SAMe and methionine work slowly. The reason they act slowly is what they do is they decrease, because of methylation, they decrease the genetic expression of these transport proteins. And so what they do is they inhibit reuptake. But that often changing the population is a process that often takes a month or two. So it can work just as well in many cases, but it's slower, and that's why it's slower. Different mechanisms. Folates are extraordinarily important in epigenetics. We now know that folates lower synaptic activity at serotonin, dopamine, and norepinephrine receptors. And this is a little bit intuitive, counterintuitive, and I'll get into that a little bit and explain that, why that happens. But we know this is true, that folates in general reduce activity at serotonin, dopamine, and norepinephrine receptors. We know that zinc and glutathione increase the activity of NMDA. That's a very important neurotransmitter. It has a lot to do with OCD, with schizophrenia, with bipolar disorder, certain forms of bipolar, and even memory. And many nutrients influence neurotransmitter activity and influence brain function. Well, turns out that epigenetics is extraordinarily complicated, and I'll give you a couple of reasons why it's so complicated. And it's probably going to take a few more decades before we really get our handle on all of this. You remember the histone proteins, where we had 8 histones that are wrapped like a ball of yarn, and all your DNA is around it, and the reactions of those histones can have a major effect on what happens. Well, today I got into the literature, and there now are 76 different histone proteins that have been identified. The last time I did this, there were 69. There's now 76 different histone proteins. Transcription factors. These are genetically expressed factors that have a DNA motif. In other words, they have the ability to attach to parts of your DNA, and these factors are needed to assist in expression of a protein. Well, there's 2,600 of them. Wow. And even in some of them, there are SNPs in these transcription factors. And then there's these microRNA molecules. These are little molecules of only involving, I think it's 22 nucleotides. They're small, but what happens is that they're around your nucleus, and say you've successfully made copied the sequence for a protein, and now you have messenger RNA that's trying to leave the cell to the nuclear membrane and get into the reticulum of the cell to make a protein. Well, microRNAs can just shut it down. If they will attach to messenger RNA and they can mess it up. So it's a very complex thing, but we already know enough to be able to help a lot of patients, and as time goes on, this is just going to explode, and I'm just looking forward to these advances in brain science and epigenetic science, because it's really going to help us improve our treatments, and I think have a lot to do with prevention of a lot of these disorders. Folates, folic acid, folinic acid, and L-methylfolate. Well, these are all effective methylating agents. Methylfolate can be the most effective if you happen to have a problem with your MTHFR. If you have a weakening of your MTHFR, methylfolate can cause the histamine to be methylated and to go back to methionine, as we talked about before. However, methylfolate is not as effective as a lot of people are believing, and one of the reasons is it's what I call a suicidal nutrient. And if you recall, those methylation cycles are constantly spinning over and over and over and over. Methylfolate reacts once, and the first time through the cycle, it loses its identity, and it becomes garden variety THF, tetrahydrofolate, and in a way it becomes part of the problem. So it can provide somewhat better functioning, but it's a short-term thing. It does its job once, and then it loses its identity, and becomes just like all the rest of the folate forms. However, there are some people who do very nicely on methylfolate and thrive on it, and there are, for example, autism. A lot of people do. A lot of the children respond very nicely to methylfolate. So it's a bit more powerful and effective than the others. However, folates increase gene expression of these transport proteins. For example, SERT, which is a serotonin transporter, or DAT, D-A-T, the dopamine transporter, or NET, which is a norepinephrine transport proteins. Folates, by an epigenetic mechanism, they increase gene expression of these proteins. And these are proteins that promote reuptake. They don't inhibit, they promote. So this explains something we've seen over the last 20 years. We've known for 20 years that if you have an undermethylated depressive who has low serotonin activity, and most of them do, they're intolerant to folates, usually. Well, let me go back to that. The reason is that if you have an undermethylated depressed person, yes, you can help them. You can improve their methylation with folate therapy, and with the methylfolate being the most effective. However, the negative effect from this epigenetic mechanism overwhelms the benefits, and reuptake dominates this so much that the improvement you get from improving methylation is just dramatically overwhelmed by the harm done by the reuptake aspect. Clues from cancer research. Most of the research in epigenetics has been done in cancer. It's really quite brilliant, and it's amazing what they've learned in a short time. They know that severe oxidative stress can alter established epigenetic bookmarks, those initial DNA methylation bookmarks that were put in there before birth. And they now know that the deviant marks, which is usually a methyl mark where it's not supposed to be, it can turn on, it can silence a cancer protective gene. They've learned that it's not so much turning on a cancer gene, but silencing a cancer protective gene, initiating a cancer condition. Just to give you an example, say a person is in the sun too much or is in tanning beds too much, and they have a lot of cumulative insults to their skin. Or maybe you've got a person who is a heavy smoker and is causing these cumulative insults to their lungs. Well, at some point, if you overwhelm the oxidative protectors, we all have wonderful antioxidant protectors in our body, whether or not we take supplements. But if you get to a point where these environmental insults are so great that you completely overwhelm your antioxidant protectors, then that can cause a deviant mark. It can cause a change in your gene expression, and therefore a epigenetic disorder. And so that is apparently how most cancers originate. And now we know that definitely know that most mental illnesses involve severe oxidative stress. So oxidative stress is extremely important. Now in the brain, especially in the brain, the first line of defense is glutathione. Well, glutathione comes from diet. It's synthesized in the body from the food we eat. And it works beautifully if there's enough of it around. The problem is that if there's a limited supply of glutathione, and actually after only 10 or 20% of your glutathione, which we call reduced glutathione, once 10 or 20% of it gets oxidized, it really loses its potency and its ability to function. And what happens then is something quite wonderful. Other proteins, notably metallothionein, are then genetically expressed, and you can have a sudden increase in the amount of this metallothionein protein. Well, metallothionein is nature's backup system. It works together with glutathione. In fact, metallothionein doesn't work effectively unless there's oxidized glutathione, oxidized, inactive glutathione. And that actually causes metallothionein to function as a really powerful antioxidant. In fact, it's more powerful at protecting against mercury than even glutathione. It's something like 1,000 times more protective. It has a greater affinity for toxic metals even than glutathione. And we know the selenium increases this functioning by about 50%. So I call these the three musketeers for antioxidant protection in the brain. And I think for so many mental disorders, probably everything we talked about, one thing that anyone could do is bolster their antioxidant situation. So I want to give a couple of examples before we finish. I'm getting near the end, by the way. Undermethylated depression. There's a lot of people with undermethylated depression. We find it to be 38% of all people that we've seen that have been diagnosed with clinical depression. That's 28% of a lot of people. We've seen 3,200 cases. So what is our approach? Well, we want to enhance methylation and suppress acetylation of histones. SAMe and methionine-active serotonin reuptake inhibitors, and we want to have reduced gene expression of SERT. Okay. We don't want to have as many of these SERT proteins, the passageways for the returning neurotransmitters. We need to avoid folate supplements, even though that will improve methylation, because it's going to cause a dramatic increase in reuptake, which is the last thing these people need. And then we use augmenting nutrients that help. And I won't get into the gory details of why, but it's zinc, serine, inositol, trimethylglycine, calcium, magnesium, and several vitamins, including some of the antioxidants. We certainly would include selenium in there. Second example, this is the last example, and you can imagine how many different examples we could give you, but this is a case of, let's say, overmethylated schizophrenia. I've seen more than 1,000 cases of overmethylated schizophrenia. And what we would do in their case is we want to support acetylation of histones using folic acid and niacinamide. We use this, and so really, the number one factor really is folic acid. Folinic acid is probably, folinic acid might be somewhat better, and methylfolate might be the best of all. But these things are wonderful at helping people who are overmethylated schizophrenics, even though it seems counterintuitive because folates are such great at elevating methylation. But they are, that and niacinamide, the folates and niacinamide are powerful deacetylase inhibitors, and that's sort of the name of the game. That is the most powerful handle we have on this. And then augmenting nutrients are things like DMAE, which is tends to increase choline in the brain and reduce dopamine activity. It's a dopamine antagonist. Zinc, selenium, chromium, and again some vitamins. And this particular therapy is especially promising for paranoid schizophrenics with excessive dopamine activity. So the whole process is a medical process. This is something you should not do at home. Those of you in the general public who are not professionals, I urge you not to try to use this information to treat yourself. You really need to go to a medical professional who's experienced in this kind of therapy. And just a summary of what it is, basically the process is not that unusual. A medical history is really important, especially the symptoms and traits that relate to the various different imbalances. There's more imbalances than just methylation imbalances. There's Pyrrole disorders, there are mental metabolism disorders, etc. Special blood and urine tests are very useful in diagnosis, and the combination of the knowledge of the patient and the lab work usually enables us to accurately diagnose chemical imbalances and therefore identify which neurotransmitters are not functioning like they should, which is an important aspect of guiding our nutrient therapy. And then a prescribed nutrient program aimed at normalizing brain chemistry. So that's NET process and the new information from new brain science and epigenetic science is really enabling us to help people that we couldn't help before and give us higher rates of efficacy. So in summary, methylation imbalances play a critical role in many mental disorders. Autism, schizophrenia, post-traumatic stress, and several other mental illnesses appear to be epigenetic in nature. Epigenetic science is providing a roadmap for nutrient therapies that can adjust neurotransmitter activity at some of the major neurotransmitters. And I just wanted to mention that I believe philosophically that as brain science advances, natural therapies may gradually, over years, eliminate the need for drug medications. I'd like to leave you with Pfeiffer's Law. For every drug that benefits a patient, there are natural substances that can produce the same effect. And I'd like to mention that for any professionals that are interested in learning these protocols, we've been training doctors around the world, and we are holding a physician education workshop in the USA, actually in the Chicago Marriott in Oak Brook, Illinois, October 18th to 22nd. And the goal of this is to enable interested doctors to learn these nutrient therapy protocols for behavior. We're going to be focusing in this particular workshop on behavior, ADHD, anxiety, depression, bipolar disorder, schizophrenia, and Alzheimer's. We're not going to include autism this time, but we plan to on the next USA program. The program has 32 American Medical Association Category 1 credits, CMEs, and for the contact information is shown there, sue@walshinstitute.org, or the phone number that is shown there. I'd also like to say that as part of this, at the same time, at the Chicago Marriott in October, we are going to be seeing real-time patients. This is a feature of our physician education program, for them to see actual live patients, many of whom we have never seen before, for whom we have the lab results in advance, and we go through the whole evaluation process, including evaluation of the chemical imbalances using, going through an in-depth medical history, blood and urine testing, diagnosis, and prescribed advanced nutrient therapy for each of these patients. So this is a way for some patients to get a really good evaluation, and there'll be a number of doctors present while we do the evaluation. And if there are any families that are interested in exploring this, the contact information is shown at the bottom of the slide. And at the same time, as part of this, since we're doing this program, the workshop over a weekend, so that the doctors won't miss too much of their clinical practice in doing this, we're doing a symposium on Sunday in the middle of this. And this, again, is on October 19th at the Chicago Marriott in Oak Brook. And it's called Nutrient Power, Advanced Methods to Heal Brain Disorders, and that is open to the general public. I'd like to show, this is the book Nutrient Power for those who want to dig into some of the details of epigenetics or of the various disorders, with their chapters on schizophrenia, depression, behavior disorders, autism, and on Alzheimer's disease. If you want to dig into that, it's available both on Amazon, but it's also available on our website. And if you purchase it from our website, whatever profits go into our charity.
Great, thank you so much, Dr. Walsh. At this time, we have some time to take some of your questions. So please enter your questions into the questions pane located in your control panel. And our first question for Dr. Walsh, this person says, I met with you, Dr. Walsh, a couple of years ago when you were in Australia. I was diagnosed with Pyrrole disorder and undermethylation. You put me on a nutrient protocol that prohibited folic acid. Recently, I had an MTHFR genetic test which showed that I was C677T heterozygous. The standard treatment is methylfolate, which is an active form of folic acid. Should I take methylfolate or not?
Well, if you're, we just covered this in quite a bit of depth in my presentation, and if you are accurately diagnosed as being having low serotonin and undermethylation, then it would be best if you not take methylfolate. The methylfolate would do a dandy job of improving your methylation, but the impact, the epigenetic impact of methylfolate or any of the folates would unfortunately would cause your serotonin activity to drop even lower. Instead of you really want to elevate your serotonin activity. Now, improving methylation will have a modest effect of improving the amount of serotonin, but that's a tiny factor compared to the massive effect on reuptake. So we recommend you don't do it. But if you do it cautiously, and as soon as you start to feel worse, we recommend you stop.
What are the most important diagnostic tests, including vitamin and mineral analysis?
Well, fortunately, there are only a handful of nutrient factors and imbalances that dominate mental health. This is completely different from the rest of the body, and there are dozens and dozens of really key nutrients that one has to consider for many of the you might say physical disorders. But for mental disorders, the major tests, for example, we know you have to know mental metabolism. So we always want to test for plasma zinc, which we know is still considered by the experts to be the most decisive measurement of zinc status in your body, although it's closely followed by Paxil studies of zinc. We think that serum copper and serum ceruloplasmin are essential, and it's great to do both together because this gives you, first of all, an idea of whether you have a copper imbalance, but not only that, it's a really nice measure of oxidative stress because by calculating you can calculate the amount of free radical or loosely bound copper, mostly copper, divalent copper, if you know the ceruloplasmin. Most of your copper is supposed to be present in your bloodstream as ceruloplasmin, but if the amount of free radical copper gets above say 30%, as you often see in anxiety and depression, or over 40% or 50% that is commonly seen in autism, that has a massive effect on brain function. So that's an important test also. Urine Pyrroles we like to have because a really high percentage of people with mental disorders have elevated Pyrroles. The number is higher than 30% for autism. For schizophrenia, it's something like 20%. For depression, it might be 15%. Behavior disorders is almost 30%. So it's something you should check for. And if, in fact, you test for urine Pyrroles and you are elevated, it's time to go out and celebrate because that is the easiest chemical imbalance to correct. It is also a chemical imbalance that can cause just dramatic mental problems. We like to do whole blood histamine, and if possible, the SAMI-SAH ratio. And we like to, so those are some of the key tests that we do, and we also want to know about liver function and electrolytes, and so we'll do a standard, what's known as a SPAC, where we'll look at thyroid and some of the other problems that can often affect mental function. But and this we find usually is all the testing we need to do. It comes to about $400 worth of testing in a panel that we can have a lab do for us, and that is usually all we need to accurately diagnose about 90% of the mental patients we see. Sometimes you get more complicated cases and you need to do additional testing. But if you did every single lab test that could provide valid information, a person wouldn't have a drop of blood left, and they probably wouldn't have any money left either. And so you have to prioritize. And the good news about mental disorders is that there are just a handful of chemical imbalances that dominate mental disorders.
What kind of practitioners do you recommend are knowledgeable in this field?
Well, I recommend practitioners that understand these protocols and have experience in it. And that's why we spend a lot of our time, we've spent a lot of our time over the last several years traveling around the world. I have a team of experts that travel around the world, and like I said, we're a not-for-profit, and our goal is to enable doctors to effectively do this, medical professionals. And so my goal is to sort of hand these protocols and give these protocols to 1,000 doctors over the next 5 years. And I think after October we'll have our first 200, so we're well on the way. And of the people that have been doing this, several of our Australian doctors have been doing this for more than 7 or 8 years. They are just so enthusiastic when they find out there are patients that they couldn't help before that are now able to help, and very often without having to resort to drug medications. We're definitely not opposed to drug medications. Drugs have helped millions of people, and I've met thousands of people who told me their lives were dramatically helped by antidepressants or atypical antipsychotics. But it would be nice if you could do it without that.
My child has an ADHD diagnosis, and tests indicated that he may be slightly undermethylated. However, his behaviors and anxiety tend to be more that of overmethylation. Are there other factors in methylation, for example, Pyrroles, which can skew the results? If so, which tests can augment?
Well, that often happens. That often happens exactly. A person may have a mild undermethylation and have a problem like a behavior disorder, and maybe meltdowns, or even academic problems. And it may have nothing to do with methylation. For ADHD, methylation is very often not the dominant problem, unless you have strong will oppositional defiance. A person like that, the first thing I'd want to know is their zinc level. So plasma zinc, plasma copper. Serum copper, plasma zinc, serum copper, and I would really like to know whether they have a Pyrrole disorder. And doing those three would probably pick up at least 3/4 of the people with bad behavior. There are some patients that are really easy to help, and others that are much more complicated and difficult. Those that are the easiest are probably young people with severe behavior problems, whether it's acting out violent behavior or oppositional defiance. Another group are females with hormonal type depressions. These are people that are especially easy to help. It doesn't take a lot of lab testing. And anyway, those are I would look at the metal metabolism and definitely Pyrroles. And then I'd also want to know about their toxic metals, because a lot of kids get in trouble if they have an overload of lead or mercury. So those would be the main things I would want to do.
Mm-hmm. Mm-hmm. You had mentioned that Pyrroles are a byproduct. What are they synthesized from, or what metabolic processes are they left over from?
Well, there are a number of reactions, natural reactions in bone marrow and in the spleen that result in Pyrroles, and a lot of these have to do with perhaps synthesis of hemoglobin and associated proteins. So that's where for some reason, and I'm sure it has to do with mutations in SNPs, some people produce just an enormously high number of these Pyrroles. On a typical, normal, healthy Pyrrole level, and we all have some Pyrroles, would be between, I'd say, 5 and maybe 10 or 15. People who are above 15 I consider to be at least borderline pyroluric, and anyone who is over 20 is severely pyroluric. And I've seen people higher than 200. I once did a study of extraordinarily violent people, including some of the more famous criminals in the last 30 years, like Charles Manson and others. And I found a serial killer in New York, in prison in New York, who had a Pyrrole level of 200. His psychiatrist asked me if there's anything we could do for him while in custody, and I showed him the protocol for fixing Pyrrole disorder, and he reported back that after a month his behavior became quite normal. unfortunately, this made the newspapers, and people didn't like the idea of a serial killer in prison getting special vitamins, so they forbade him getting these, and so he's in trouble again.
Mm-hmm. Is it possible to be an undermethylator in one part of the cycle and an overmethylator in another part?
Well, the cycle itself, if you're talking about the methylation cycle, the only part of the cycle that might tend toward overmethylation would be the cystathione part, where you're creating these sulfur antioxidant proteins, because then part of the homocysteine would be going to something other than the methylation cycle. But in general, you would wind up with undermethylation if there was a problem there. The overmethylation mostly comes from the SAMe utilization. In other words, what happens to the SAMe that's produced in that methylation cycle. So you really don't have both at the same time.
Some experts say that serotonin is made in the gut. You had said that serotonin and dopamine is made in the brain. Can you please explain?
Oh, yes. Most of the serotonin in your body is not made in the brain, and a lot of it is made in the GI tract. However, all of the serotonin that in your brain is made in your brain. None of the serotonin in your stomach gets to the brain, or maybe a couple of molecules, but virtually little or negligible amounts ever make it up there. So really, it's the synthesis in the brain with respect to serotonin, dopamine, and some of the other neurotransmitters that matters. And a lot of people misunderstand that. You can actually have quite different levels of serotonin in the body as compared to the brain. And in fact, the synthesis, there is one mechanism in the brain synthesis that is quite different from what happens in the gut. So they're quite different. So actually, if trying to adjust your serotonin situation in your gut may or may not have anything to do with your brain problem.
How does one prevent glutathione oxidation?
Well, we all have glutathione at the ready that is protecting us. And the number one way that it gets depleted and not available to do what it needs to do is oxidative stress. And so for example, let's say a person has exposure to mercury or lead. That will deplete your glutathione. Let's say you have an immune disorder, and that can cause oxidative stress. So what happens is that you've got basically a war between the good guys and the bad guys. The good guys are your glutathione and other natural antioxidants. And then the bad guys are all of these factors that can cause oxidative stress. Even includes things like nuclear radiation and numerous illnesses and numerous toxics. It might be even pesticides. So when you're in a bad environment and the body is assaulted by too many or excessive amount of these oxidative chemicals, then you wind up with too many free radicals. The glutathione, which has there's a limited amount of it there, a limited supply, you can use it up excessively, and that's what happens.
What is the best way to enhance the methylation cycle in a person who shouldn't take folate?
Well, you're really limited there, unfortunately. Certainly B12 and methyl B12 would be good. Another really good way is trimethylglycine. And then, of course, you have to make sure that you have sufficient methyl itself and sufficient methionine. You can help it a bit by additional methylation. I want to mention one point that I think I neglected to say. When we're talking about folic acid having a negative effect on patients, we're only talking about mental patients, patients with brain disorders who have low serotonin activity or low dopamine activity. Methylfolate is probably dramatically the best way to help everybody else. So we're only talking about mental disorders involving neurotransmission problems. And all these other people Folate therapies, and in particular methylfolate, should work just beautifully and do work beautifully. An example is autism. I've worked with many autistics, and very few of them have neurotransmitter problems that are a major part of their issue. So they don't have to worry about the epigenetic problem of folates. They can thrive on methylfolate or the other folates. And very often folic acid itself can be really effective together with B12 for these people. So I'm glad we had a chance to at least make that point. Methylfolate is a clever way to help people who have really serious SNPs in their MTHFR.
Do you feel that having only one MTHFR mutation, such as A1298C, needs attention, as in folate, B6, and B12 in higher dosages?
Well, everything I've read, and there's a lot of research on this, what I've been reading says that if a person is heterozygous in MTHFR for the 1298, which is probably the second most significant location for a SNP, that it generally has very little or no effect on the enzyme itself. Mm-hmm. and those people very often do not require therapy of any kind related to methylation. One way you can sort of evaluate the seriousness of a SNP like an MTHFR is to have a homocysteine analysis. If you have an MTHFR problem and you also have elevated homocysteine, then that's something you really need to address. But very often, like in autism, we have almost all autistics are undermethylated. The great majority have MTHFR SNPs, and they're undermethylated, and it's just not a problem for them.
Dr. Walsh, another methylation expert, has said that once your glutathione cycle and glutathione levels are addressed, that no other methylation cycles need to be addressed. Is that your view too?
No, I think there are a lot of things that can go wrong, and he's absolutely right about if your glutathione cycle, which is really the cystathione pathway, if that's not functioning, yes, you're going to be in terrible trouble. I agree with that. But there are a lot of people who can have adequate antioxidants and still have severe methylation problems, and I think epigenetics really explains that and explains why and why it can be so powerfully important. So if you've got two possibilities of terrible things happening in your methylation cycle, one is too little antioxidants, too little glutathione, and the other is too little SAMe available to do all those important reactions in the body.
What role does epigenetics play in autoimmune disorders such as Hashimoto's and Meniere's disease?
Well, there's been suggestions that a lot of these physical diseases are epigenetic, and one would have to look at those criteria that I mentioned. I've not personally looked at either of those, but I think that researchers and medical experts are going to start looking at all of these disorders, especially those that appear rather suddenly, where a condition that didn't exist in a matter of weeks or maybe a month or so suddenly appears and then is refractory, meaning it's really difficult to change it. I imagine there are people, epigenetic experts, who are doing this and just sort of going through every medical disorder that exists, physical or mental, trying to identify which ones might be epigenetic. Because if they are epigenetic, that should lead to more targeted research, better understanding of what it is, and then may give a roadmap to effective treatment.
Dr. Walsh, there are often doctors who prescribe biotin for patients. What's its role in mental health?
Biotin is a really interesting nutrient. For a while, for about 15 years, when I was working with Carl Pfeiffer, we would give biotin to slender malabsorbers. It has a really major impact on GI tract function, and in fact, biotin is synthesized in the GI tract. But biotin also is one of the major nutrients that's involved in epigenetics, and we haven't really worked out all the details. But biotination is alongside methylation and acetylation in affecting histones and affecting genetic expression. So it probably is far more important than we realize. But we also know from research done by Woody McGinnis, Dr. McGinnis, we know that that people with Pyrrole disorder are deficient in biotin. So that's another example of lab work that can help tell you something. There's an inverse relationship between Pyrroles and biotin. So we might want to give quite a bit of biotin to Pyrrole patients. In fact, we do, because we know they need it.
Dr. Walsh, how do you test for heavy metals?
That's quite difficult. Heavy metals are somewhat difficult to work with. I've been working and studying metal metabolism for a long time. It depends on the metal. For example, mercury. Mercury can do terrible things in the body and in the brain if it can make it into the brain. We have a really elaborate system for protecting us against, let's just look at mercury, for example. We know that in the GI tract there is quite a bit of metallothionein and some zinc that can disable some of the mercury. In other words, react with it and make it safe. And the intestinal barrier, one of the key parts of the intestinal barrier is metallothionein. It's believed that 90% of the mercury from your diet, let's say, that tries to make it into the bloodstream is stopped by metallothionein. Metallothionein is like a sponge for mercury, and then the mercury then would go to the liver in the portal bloodstream, and again you would have glutathione and you would have metallothionein in the liver, and they think about 90% of whatever mercury made it that far would then be made inactive by reaction. And then the tiny bit, the 1% or less that might try to make it to the blood-brain barrier and get into the brain, again you have metallothionein, which is the #1 barrier for metals in the blood-brain barrier. And so if all those systems are working, a person would take probably a fairly significant mercury exposure to harm them. But there are people who don't have those protections. There are people with very low glutathione levels and very, very low metallothionein levels, such as autistics, for example. We know their metallothionein protectors are at very low levels. I was the first person to ever test metallothionein in a human being. And we did this with autistic kids, and we found a clear correlation. They're low in metallothionein. So what does that mean? That means they're far more sensitive to mercury. So if a person has high toxics and it happens to be mercury, probably the best thing you could do would be to do whatever you could to improve the genetic expression of metallothionein, or provide glutathione, however you would like to do that. It's not easy getting glutathione orally into the body because glutathione is made up of three amino acids, and they don't survive the GI tract, so they break back into these 3 amino acids, and so it's not easy to improve glutathione levels in your bloodstream and in your brain orally. So that's why people are using clever ways of trying to get it into the body, such as liposomes, where you have fatty substances bound to glutathione, and this is appears to be quite effective. There's also transdermal glutathione that you can put on the skin and get glutathione directly into the skin. Well, that's mercury. One of the major toxic metals is lead. The problem with lead is that the half-life in the body is 22 years. 22 years, and the reason is when lead gets in the body, it thinks it's calcium, and it gets reabsorbed into your bones, into your skeletal structure. And so if you've had a long-term exposure, your long-term lead, a lot of it's stuck in your bones, and it's going to be leaving very gradually. So if you're lead poisoned or have a lead overload, you have to continue to do antioxidant therapy really the rest of your life. The half-life of mercury for most people in the body, in the periphery of the body, is 42 days in healthy people, and in the brain it's 70 days. So mercury can do a lot of damage, but at least it tends to leave the body rather readily. I did a study that I haven't finished, that I have not published yet, where I actually was examining, measuring mercury levels in brain tissues from an autism brain bank, where we had brain tissues from autistic children and from typical children, both who have died at an early age of something else, and looking for mercury to see if, and now each of these kids that we had looked at, the autistics, they all had thimerosal, so they were all exposed to mercury. And so basically our conclusion is that it looks like mercury could very well trigger autism if enough of it got into the brain or into the body. However, it looks like it doesn't stay in the body and cause continuing harm, at least not in the brain.
Dr. Walsh, you talk about methylation and epigenetics in relation to many mental illnesses except eating disorders, in particular anorexia. Has there been any research in connection to eating disorders? Absolutely, yes.
Anorexia and bulimia both are undermethylation disorders. And when you talk to some of these patients, these are conditions that can suddenly appear. Had a lot of teenagers who did not really have evidence of either of these disorders, and it just seemed like they started all at once. And they do also somewhat run in families. They're heritable. They certainly don't follow genetics. There's a lot of indications that they may very well be epigenetic. But the one thing that makes me think that they're not epigenetic is that they're correctable. I've had so many cases of anorexics and bulimics who got completely better. And that doesn't usually happen with an epigenetic disorder. Epigenetic disorders usually do not go away, at least they don't go away very easily. And they usually cause problems the rest of your life. We've had hundreds of anorexics who, most of them were young people. And the combination of nutrient therapy, especially methylation therapies, and counseling. We found that counseling is a key part of it too, but that combination usually can make the anorexia or the bulimia go away, and very often go away forever. So that tells me that probably is not epigenetic.
What kinds of results have you had using targeted nutrient therapy for treatment-resistant depression?
Treatment-resistant depression is depression that that's a form of depression that apparently does not involve low serotonin activity. The psychiatric world is suffering from a misconception with respect to depression, and throughout the entire world, if a person is diagnosed with clinical depression, the belief is that these people have low serotonin activity, and that they need to take a medication that is a reuptake inhibitor. Well, the people who are resistant to reuptake inhibitors, these are the people who are overmethylated. They have too much serotonin activity to begin with. These are the people that get dramatically worse when they take an antidepressant. Many of these are considered to be what they call in psychiatry serotonin syndrome patients. They can be helped very nicely, very easily. In fact, we found a lot of these people who had failed with one mainstream therapy after the other, and they had taken 5, 6, or 7 different antidepressant medications. We've had reports of great success. We've done outcome studies for these people, open-label outcome studies, and our success rate is actually higher for them than with the more typical low serotonin depressants who actually may do well on antidepressants, perhaps with side effects that they hate.
This is going to be our last question for the evening. Once a patient with depression has gone onto the appropriate protocol, vitamins and minerals, and the depression has lifted, how long before they can go off of the protocol, or do they need to remain on the supplements permanently?
Well, so far we have the ability to treat problems like depression, but we don't have a method yet of curing it. Although I think in 10 or 20 years, epigenetics will advance to the point where that will be possible. When most people we see who come to us with clinical depression are taking an antidepressant or some other medication, we insist they stay on the medication. We've learned through from long experience that our patients, we have a higher and more rapid improvement in patients if they continue the medication and do the nutrient therapy, do both treatments together. Then usually after about 3 or 4 months, for those patients who do really well, and a lot of them do, most of them do, based on the reports they give us, and occasionally we've done Beck scales and Hamilton D scales to measure the before and after level of depression. Then after we see the improvement, we suggest they go back to the doctor managing their medication, and they cautiously try lower and lower levels of the antidepressant medication. And what they tell us is that 80% of the depressive people say that they can go to 0. The other 20% say they lose something if they try to eliminate all of the SSRI or whatever the antidepressant is. And we say, so be it. We are not opposed to medication. We just want the patient to be at their best, and if possible, with the minimum amount of side effects.
Thank you all for joining us this evening for our special presentation. Please support the Walsh Research Institute nonprofit organization by purchasing a copy of Nutrient Power on our website, walshinstitute.org. 100% of the book proceeds go towards our organization. And please, if we didn't get a chance to answer all of your questions because we had so many participants this evening, please feel free to send your questions to Sue, S-U-E, at walshinstitute.org. That's Sue at walshinstitute.org. And thank you again.